Hey friends, I got a slightly odd request. I was asked to explain my thesis research. I'm old, this was a long time ago, so my first thought was, it's so long ago and it's so irrelevant. Why would you want to learn about ancient history? And then, worst of all, my second thought was, but I no longer think my hypothesis was entirely valid. How am I going to explain that science advances? And then I realized that maybe this isn't important to make. And it might be good to know that a lot of science is about flailing around, trying to figure something out, and that it's universal and timeless, even if it was done in 1985. So here's the story. As a naive young student, I entered the lab of Charles B. Kimmel at the University of Oregon, where the interest at first was about how the Mouthner cell works in embryonic and larval zebrafish. It would later change and evolve. That's another lesson. Nobody does the same thing forever. Okay, so that's me. The more distinguished looking gentleman is Chuck. And okay, I might have been a little older than that when I started grad school, but not much. It certainly felt that young. Okay, so I better explain what a Mouthner cell is. So what is a Mouthner cell? We're looking down on a fish. There's two big eyes. There are ears just behind them, and inside the hindbrain is a pair of giant cells, the Mouthner cells. These have been known about since the 19th century when Ludwig Mouthner discovered them in 1859. And that was a time when if you found a new kind of cell, you just name it after yourself. It's cool because it's what is called an identifiable neuron. You can see this cell in every fish, every amniote actually, since it's also in amphibians, and you can study its properties in detail in multiple individuals, finding the same named cell in every single fish. This is unusual in us vertebrates, but common in invertebrates. They're also interesting because they're involved in a simple, specific behavior. They receive input from the eyes, the ears, and the lateral line, sensory modalities in general. So if something abrupt happens, they sense it, and the Mouthner sends a massive signal down the spinal cord, activating all the motor neurons and triggering a fast contraction. The fish arches away from whatever startled it. This is called the startle escape response, and you can see it when you tap on the glass of an aquarium. The fish all abruptly jerk away. Let me just say, it's mean to do that. Don't do that. Why do you want to startle the poor fish? Okay, so I started out in the lab poking baby fish in the ear and recording the extracellular potentials generated by the Mouthner cell. It's a huge cell, hundreds of microns across. It generates large currents and voltages, and you can even record its electrical activity by just placing an electrode in its vicinity. So it's a little bit like mind reading, that you can at least read this one cell just by recording the currents that flow outside of the cranium. The physiology of the Mouthner cell is heavily researched, and even though it's a single cell, it's complicated. Now, you might be wondering if you have a Mouthner cell and you don't. Think about it. A big jolt that causes one side of your body to spasm is an effective way to get a tail to snap and fling to propel you out of danger in the water. But in terrestrial organisms, it would just make you fall down and thrash around, kind of the opposite of escape. This cell is part of the reticular formation, though, and you do have that. The reticular formation is a grid of fibers and distributed nuclei that process sensory information in you and play an important role in general alertness. When someone surprises you by tapping on your window, you don't throw yourself across the room, but you do snap to attention. Your brain gets to work trying to figure out what made that noise. I include this here just to tease the Jordan Peterson fans who seem to think serotonin, that is 5-HT, is the only neurotransmitter that counts. Mouthner has all kinds of sensory inputs and all kinds of neurotransmitter receptors, and it's got all kinds of intricate modulation. See that thing labeled axon cap? It's a tangle of fibers that come close, but don't touch the cell. They generate a response with field effects. The whole thing is kind of mind-blowing. And remember, this is one cell in the brain of a simple fish. But I didn't want to do physiology, fun as it was. When I entered the lab, Chuck had just made an amazing discovery. Chuck's lab was figuring out the detailed morphology of the Mouthner cell. To do this, they used a tracer enzyme called horseradish peroxidase. You come in with a needle or a knife coated with HRP at some distant point on the axon that you don't care about, and you basically make a hash of everything right there. And the enzyme is then transported through the cell, and you can stain for it, see every little dendrite and bump and bulge in the cell with high contrast. The only thing you can't see is that terminal end where you injected all the HRP. Kimmel and his students had done all this before I got there, and they had a surprise. When they put HRP into the spinal cord, they lit up a small collection of cells. Not just Mouthner, but serial homologs of Mouthner, all part of that structure called the reticular formation of the hindbrain. And they also labeled some vestibular nuclei cells that project into the spinal cord, and cells of a structure called the nucleus of the medial longitudinal fascicle. They found something like 27 different classes of cells that project into the spinal cord, and 19 of those were in individually identifiable pairs distributed in a precisely segmental arrangement. On the one hand, this means the problem of figuring out the behavioral circuitry has gotten much more complicated. That's a common phenomenon in science. On the other hand, we'd suddenly gone from one individually identifiable pair of cells in vertebrates to almost a score. So many exciting things to figure out. I can't emphasize how cool this was. Lots of identifiable neurons and specific circuits meant this system could be taken apart component by component. And in addition, their arrangement was this elegant ladder where similar components were repeated in a segmental fashion. It was looking like a fruit fly in some ways. And yeah, later those segments were found to be defined by specific Hox genes. Look at those beautiful stripes of Hox expression in the hindbrain. But I'm getting ahead of myself because this was work yet to come, and I'm not going to have any part in it. But when I entered the lab, they just worked out this elegant system with tons of questions to be worked on. It was a good time to amble into the lab as a student and get asked, okay, what slice of the pie do you want, kid? And like a fool, I ignored all the potential great projects in this elegantly discrete set of cells in the hindbrain, and I asked instead, what's going on in this mess that you've made in the spinal cord? Remember, with HRP, you make a hash of a lesion site with a needle or a knife, or often in zebrafish, you could just dip scissors in HRP and slice off the tail and leave an obscuring smear of the enzyme there. So I wanted to know what was going on in that dark blob down there. Okay, it wasn't actually a foolish question. I was personally more interested in development than in physiology or behavior, and I was approaching a classic problem in development. How do migrating cells navigate to a specific destination? Why does neuron A connect to neuron B but not to neuron C? We need to figure out what the targets are in the spinal cord. Who is the Mouthner cell connecting to? Unfortunately, we knew nothing about the zebrafish spinal cord at that time. But we did know some general facts about spinal cords in simple vertebrates. We knew a fair bit about the general properties of the vertebrate spinal cord. There was some great stuff from the first half of the 20th century by George Coggill and Charles Judson Herrick, who studied the development of the spinal circuitry in salamanders. They proposed that there was an early primary circuit made up of large neurons that regulated whole body behaviors, like the startle escape response, and that also controlled the oscillations of swimming. They identified cells they called primary motor neurons in amphibians and assembled hypothetical circuits of cells that would set up wave-like undulations of the body. I was also inspired by papers from the 1940s and 50s by Silvano Leghisa, in which he used silver stains in trout to find Herrick and Coggill's primary motor neurons in those animals, and also identified a smaller class of cells with smaller peripheral fields that he identified as secondary motor neurons. He argued from morphology that the Mouthner axon selectively made synapses with the primary motor neurons. Groovy, so I had a good reason to suspect a two-tiered arrangement of motor neurons in the spinal cord, and I went to my advisor and my committee and proposed to map out the motor neurons of the zebrafish spinal cord. And I would use this familiar technique of HRP staining to do it. It was simple. Okay, if you look at this diagram, there's this central cylinder in the larva or the embryo called the notochord. And there are 4 quadrants of muscle around it. And they also wrap around the spinal cord, SC up there. In that little diagram, you can see there's a dorsal fin, dorsal median septum, DMS, that separates left and right. There's a spinal cord. There's the HS, or horizontal septa, which divides the muscle into dorsal and ventral quadrants. And something called the VMS, the ventral median septum, which again is another connective tissue membrane that separates left and right. So, my plan was I would go in with a dirty needle covered with HRP and mess up regions of muscle. Then the cells that sent a process through that damaged region would pick up the enzyme, and I'd be able to stain their cell bodies and see where they are and what they look like. Okay, that's simple. That's my protocol. That's all I was going to do for the first part of my thesis. And what I was going to end up with is an anatomical map of the spinal cord. Now, wait a minute though. You also need a hypothesis to do science. I had a few ideas there too. So there were a few things we knew about the development of the spinal cord. We know that it generally grows from the inside out. So new fibers grow on the outer surface, and then as newer fibers emerge, they grow on top of the earlier fibers. So older processes are deeper, newer processes are more superficial. I predicted that the axons of primary motor neurons would be earlier and deep, and that secondary motor neurons would grow out later, and that we'd be able to see that arrangement in the spinal cord. Spoiler, this is going to turn out to be true. Okay, so that's part of the hypothesis. We got a temporal order of development that we could detect. Furthermore, I proposed a simple timing mechanism for how the Mouthner cell would selectively innervate only primary motor neurons. So primary motor neurons would grow out first. They'd grow out early. So they'd send a process out into the periphery. Then the Mouthner axon would descend from the spinal cord. It would grow down the ventral fascicle of the spinal cord. And as it grew, it would see the primary motor neurons as it passed by, and it would connect to them. So it would only connect to the primaries because the secondaries hadn't developed yet. And it would just keep going down the spinal cord, connecting to all the available primary motor neurons. And then later, the secondary motor neurons would come along, long after the Mouthner axon growth cone had passed by, and they'd grow out into the periphery more lateral to the Mouthner axon. Another spoiler, this would turn out to be true too. So this observation would be compatible with the idea that Mouthner could just use timing to find its targets. So it didn't need any specific information other than the timing of development. Unfortunately, I would say now, with the benefit of hindsight, it's doubtful that it's the only mechanism because there's other data that's going to emerge later. And it's going to say there's other additional factors that have to play a role in setting up this system. Okay, got it? So that was my thesis proposal, to map out spinal cord organization and test the hypothesis that the timing of development might be sufficient to constrain connections in the circuitry. So I did it, and short answer, everything worked as planned. So this is a cross-section through the spinal cord of a larval zebrafish. And what you can see there is on the two sides of the spinal cord, you can see these great big black cells. Those are primary motor neurons. I could see them quite clearly. And I will also say, to my own credit, that the histology I did back then was just gorgeous. I earned my Ph.D. just with with good work like that. Anyway, so there's the primary motor neurons, and you can see that they grow down, they grow ventrally to the bottom of the spinal cord, and they wrap around the mouth nerve cell, which is labeled with an M there. So right away I can see, yeah, primary motor neurons have axons that are more medial than the mouth nerve axon. Therefore, we would assume that the primary motor neurons came first, and then mouth nerve came cruising along later. Also, in the inside of this, you can see that terminal bouton that mouth nerve makes on primary motor neurons. So we also knew that mouth nerve was connecting to those primary motor neurons. Yes, also, secondary motor neurons appeared later. They sent axons lateral to the mouth nerve, as I predicted. It took only a few years of stabbing fish and slicing them into thin slabs and staining them. "Gimme my Ph.D., I said. Except, of course, I stirred up more questions, because all science does that. You get more questions all the time. My results initially showed that there were roughly 3 primary motor neurons in each segment. So I knew something about the cell bodies, but remember, HRP staining involves messing up some part of the projection of the neuron, so I didn't know much about the connectivity these cells made in the muscles. I just had dark black cell bodies and dark black muscles. Interestingly, in this image, you can see 2 primary motor neurons only. You see, if I poked the ventral muscles, filled them with HRP, the enzyme would be transported back to 2 primary motor neurons. If I poked dorsal muscles, then the HRP would get transported back to a single primary motor neuron. So it looks like a division of labor. There's 3 primary motor neurons, 2 of them project ventrally and 1 projects dorsally. In addition, when I looked at the nerve fibers, so that's what you see in the 2 panels on the right, what I saw was in the dorsal nerve tract, there would be 1 thick, prominent fiber from the primary motor neuron that went there. And in the ventral tract, there would be 2 thick fibers and a bunch of smaller ones for the secondary motor neurons. It was suggestive, but I needed to dig deeper. And as I said, there were some limitations to this HRP technique. But just then, Chuck Kimmel had worked out a brand new technique for labeling cells, and before he'd give me my degree, he thought I should try it out. And to make it less like throwing me into a whole new project, he teamed up with a new postdoc in the department, Judith Eisen. Judith Eisen would do all the hard work. So thanks, Judith. It was great. You helped me to graduate. Okay, so what is this technique that we're going to try? It's something completely different from HRP states. So we'd start with a blastula stage embryo, just a ball of cells. So that's what I'm kind of interested. That's my illustration here. There's this darker yolk down below, and over it is this ball of cells. There's thousands of cells in there. We typically worked at around the 1,000 cell stage. So we pull out an embryo at this very young blastula stage. And then we, or in this case Judith, would go in and delicately pierce a single cell with a tiny glass hypodermic needle and inject it with a dye called rhodamine dextran. This was a bright red fluorescent compound coupled to a large sugar, so it would stay in the 1 cell we injected. Then we'd sit back and wait. So we got 1 cell labeled bright red in this embryo. And that cell would do what all the cells do in the blastula. It would divide. And it would divide some more. And it would divide a bunch more. This is called lineage tracing because all the descendants of the original cell would be labeled with the red dye. So we get a little clone of cells that are all related to each other through that first cell we injected. And then, because of the nature of the way cells shuffled around in the blastula and gastrula, those descendants would be scattered throughout the embryo. So we'd have single cells sprinkled throughout, sometimes in clumps, sometimes individually. And we'd come in the next day when the animal's 18 to 24 hours old, and we'd look, and we'd find these lovely little fish-shaped embryos with glowing red cells semi-randomly sprinkled throughout. And in some small percentage of cases, a few percent actually, by chance we'd see an individual primary motor neuron labeled. What was awesome about this is that we'd label it before it had even made an axon. So we could watch it in real time as it grew out, left the spinal cord, and grew along a pathway to innervate muscle. And of course, we could also see which cells innervated which muscle fibers. It was beautiful stuff, really elegant and pretty. And we also sometimes labeled secondary motor neurons. In this particular illustration, we, by chance, labeled all three primary motor neurons at once on one side of the embryo, while on the opposite side, we'd labeled a little cluster of secondary motor neurons. We could watch and directly compare their growth, and yes, we saw with our own 4 eyes that secondary motor neurons grew out distinctly later than primaries. So it was a secondary confirmation of what I'd proposed earlier. Now, in this illustration, we've labeled all 3 primary motor neurons at once. You can't really tell who is who. But in other cases where we labeled individual primary motor neurons, we could look and figure out who was precisely innervating what muscle fibers. So we learned something surprising. Those 3 primary motor neurons in each segment, they were individually identifiable. They had an attribute just like the old mouth in a cell, and that you could go back to these cells over and over again in different individuals and see what they do. And they were predictable. The one that was closest to the ventral root would always grow out, die ventrally, and branch within the ventral block of muscles. We named it CAP, for caudal primary. The next most anterior one would grow a short distance to the ventral root, slide out of the spinal cord, and then turn dorsally to innervate the dorsal block of muscles. And we called that one MIP, for middle primary. Then the anterior-most one, which had to grow the longest distance within the spinal cord to reach the ventral root, would emerge and branch into the gap between CAP and MIP peripheral arbors. That one we called ROP. You can see a little more clearly why they got those names when we stack them on top of each other. So the one in red is CAP. It's always right near the ventral root where the nerve exits the spinal cord. And MIP is just a little bit anterior to that, and further still is ROP. And ROP is in green in this diagram, and it innervates this middle tier of muscles, while CAP innervates the ventralmost, and MIP innervates the dorsalmost. So now we also had a set of identifiable neurons in a spinal cord as well as a hindbrain. I scribbled up these stories and published them, and that was my thesis. I was done. I got to graduate. Looking back on it, was I right? Did I find the answer to the problem I was trying to answer? No. I found an answer that was compatible with the evidence. We discovered some cool stuff about the anatomy of the spinal cord, but we also stirred up new questions, like always. So, you can ask, what defined the identity of motor neurons within a segment? They clearly weren't the uniform group of equivalent cells I expected. What signals inform the cells connect to specific muscle groups? Furthermore, there was work going on at the same time in John Kawada's group at the University of Michigan to puzzle out the inner neurons in the spinal cord. The Malthus cell was growing into a series of circuits, not just an indiscriminate mass of cells, and making specific and different connections to different cells in the spinal cord. Timing is not sufficient to explain this degree of specificity. So we still had the question, how does it sort everything out? I didn't know. We still don't know. Well, there's all kinds of new data coming out all the time. Also in the 1980s, there was this amazing work being done in insects. In insects, they were identifying molecular markers on the surfaces of cells of the nervous system, identifying diffusible signaling factors. And no, my parsimonious explanation they just decide what cells to synapse on with the basis of timing was not going to work. It was not sufficient. There was another remarkably precise and specific layer of information in insect cells, as well as in fish and human brains, and I hadn't even looked at that. So after I graduated, I went off to do a postdoctoral stint playing with bug ganglia, but that's a whole different story. I think I've talked long enough now, so I will just end there, and maybe some of the time I'll talk about my postdoctoral work on grasshoppers. Okay, thanks. Talk to you all later.